Imagine trying to fix a typo in a 3 billion letter book while wearing oven mitts. That's what gene editing has felt like for years. Well, today, the oven mitts are coming off. The first fully AI-designed gene editor is here, and it's fundamentally changing how we edit our own DNA. Here's the core issue. CRISPR has always been like a GPS combined with a pair of molecular scissors. It tells you where to go in the genome, and then it makes a cut. But choosing the right guide RNA, those GPS coordinates, was a total guessing game. You might hit the perfect spot, or you might accidentally snip a perfectly healthy gene. It meant months of trial and error, and most of the time, the results were underwhelming. So, researchers tried something different. They fed a neural network a massive feast, the data from over 80,000 genomes and millions of real-world CRISPR experiments. The AI started seeing things we couldn't. It spotted subtle patterns in the DNA letters, these hidden rules shared by all the guide RNAs that actually work really well. And the result? The AI model can now predict which guide RNAs will succeed with about 90% accuracy, and it does this before a single scientist ever touches a test tube. That just wipes out a huge, frustrating chunk of the guesswork. But what about those dreaded off-target cuts when the CRISPR scissors snipped the wrong address? The AI analyzed 55,000 of those mistakes. It learned where they're most likely to happen across the entire genome, and it basically says, nope, skip that one. It's like turning off a chaotic autocorrect that keeps changing the word gene to genes. But here's where the story takes a turn. The team didn't just stop at making predictions. They built something new. Something called OpenCRISPR-1. This is the first gene editor designed from the ground up 100% by an AI. And when they tested it in human cells, it actually outperformed the natural CRISPR systems we've been using for years. This AI isn't just about scissors either. It can optimize base editors. Think of them as molecular pencil erasers that can flip a single DNA letter without even making a cut. And it can fine-tune prime editors, which are like the find and replace tool for our genome code, swapping entire phrases with surgical precision. So why does any of this matter? Personalized gene therapies. Imagine designing a treatment that's tailored to your exact genome, with an AI predicting precisely how your unique DNA will respond and steering clear of risky detours before you even start. Think of it like this: a digital twin of your genome running on a supercomputer. Before any doctor touches a single cell, the AI can virtually test millions of guide RNAs, base edits, and prime edits. It maps every likely off-target mistake and automatically reroutes around them. It's a flight simulator for gene therapy. Months of lab work, all that guesswork, it collapses into an instant, personalized game plan with a clear confidence score. The risky paths are just crossed out in red. This isn't trial and error anymore. It's laser-guided navigation through the code of life. Now, yes, this is powerful stuff, and that's exactly why safety and ethics are non-negotiable. These tools are used under strict lab protocols, and any real-world therapy will face incredibly rigorous trials. But the direction we're headed is clear. Biology is shifting from a game of chance to something predictive, precise, and deeply personal. If CRISPR was the Swiss Army knife of genetics, AI just added the laser-guided instruction manual. Fewer mistakes, faster breakthroughs, better editors, and more lives we can potentially change. It's a little scary. It's also absolutely exciting. We are witnessing the moment where computer code begins designing genetic code. So I'll leave you with this question. What would you want a tool like this to fix first? A rare disease? A pathway for cancer? Or something we haven't even imagined yet?